In our definition of life, we said that all living things are made up of cells. Knowing some basics about cells can help us understand how living things function. In this class, we will talk about those functions on multiple scales, from looking at the whole organism to looking at what's happening on the cellular level. To understand these cellular processes, we need to understand the basic makeup of a cell, what its different parts do, and how it regulates its internal environment. One of the most important structures for this regulation is the plasma membrane. The cell's plasma membrane has many different functions. First, it encloses the cell's contents, keeping them separate from the outside environment, similar to how our skin encloses the rest of our body and creates that barrier between internal and external. Also, like our skin, the plasma membrane can regulate what comes in and what goes out of the cell. The plasma membrane is semi-permeable, meaning particles and molecules can pass through it. We'll talk about that more in a minute. But not just anything can pass through, and the cell has some ability to regulate what enters and leaves. This allows the cell to take in food and nutrients through its plasma membrane. The plasma membrane also allows for the building and exporting of molecules. In a minute, we'll talk about proteins in the plasma membrane that help transport molecules and particles into and out of the cell. This allows the cell to get what it needs to build molecules and to excrete certain molecules. Some of these proteins also function as enzymes, catalyzing reactions in which molecules are built. And finally, the plasma membrane allows the cell to interact with the environment and neighboring cells. Some of those proteins embedded in the membrane are receptors that are designed to bind to specific molecules. This allows the cell to recognize chemicals in its environment and respond. So let's zoom in on the structure of the plasma membrane. Its basic structure is a phospholipid bilayer. Bi means two, so the plasma membrane has two layers of phospholipids. Let's zoom in a little more and see what that phospholipid looks like. Phospholipids are made up of two parts, a head and a tail. The head is hydrophilic, meaning it is attracted to water. It's composed of a glycerol and a molecule that contains phosphorus, which is where the phospho part of phospholipid comes from. The tail is hydrophobic and repels water. This is the lipid part of the phospholipid, and lipids are composed of chains of carbon and hydrogen. The two layers are arranged so the hydrophobic tails face each other, and the outer lining of the cell and the inner lining are the hydrophilic heads. The external environment and the internal environment of the cell are usually aqueous or made up mostly of water. So the hydrophilic heads of the phospholipids do not repel that water. However, because the hydrophobic tails in the center of the membrane do repel water, they help create a stable barrier against most water-soluble substances. This allows the cell to be selective in what enters and exits through the plasma membrane. Pause the video now to answer this question from participation assignment 1. So how does the cell allow molecules to pass through the phospholipid bilayer? As we mentioned before, there are also proteins embedded in the plasma membrane that can have many different functions, such as transporting molecules into and out of the cell, or detecting molecules outside the cell by serving as receptors. So the membrane actually looks like this. We have our phospholipid bilayer with many different types of proteins and other molecules embedded in it. These proteins can have many different functions for regulation and sensing and communicating with the environment. One of the other components of the membrane is cholesterol. We'll talk more about cholesterol when we talk about the circulatory system and the heart. You may think of cholesterol as bad because too much of it can lead to heart disease. But we actually do need cholesterol to maintain the flexibility of our cells' membranes. Remember that the hydrophobic lipid tails repel water, preventing water and the substances dissolved in water from passing through. But the cell needs to move molecules in and out. Transport proteins provide a channel through the membrane for these molecules to pass. They can be opened and closed to regulate that flow into and out of the cell. But these are only for water-soluble molecules. Some molecules that are not water-soluble, like lipids and oxygen, can pass directly through the membrane because they are not repelled by the phospholipids. Receptor proteins allow the cell to sense and respond to the external environment. Each receptor is structured to bind to a very specific molecule or set of molecules. When molecules bind to receptors, it triggers some kind of response from the cell. This allows the cell to identify the chemicals surrounding it and respond. Similarly, recognition proteins and carbohydrate chains allow cells to recognize each other. Recognition proteins on one cell can bind to carbohydrate chains on another cell, allowing them to recognize and adhere to one another. And finally, we also have enzymes embedded in the plasma membrane. Enzymes are molecules that catalyze reactions. This means they facilitate or speed up a chemical reaction, such as when the cell is trying to build a particular molecule. We'll talk more about enzymes in the lab for this class. But what's important to know about enzymes is that they are specific to a particular reaction. They work like a lock and key, where the substrate or substrates, the molecules going into the reaction, bind to specific locations on the enzyme. This allows the enzyme to facilitate or speed up the reaction by stabilizing the substrates or holding them in a specific position so that the reaction can occur. Pause the video now to answer this question from Participation Assignment 1. Let's talk a little bit more about how molecules move into and out of cells. We mentioned that some molecules can pass directly through the plasma membrane, and others pass through transport proteins embedded in the membrane. This can happen either passively through diffusion or using energy through active transport. To understand the difference between the two, we need to talk about what a concentration gradient is. A concentration gradient exists when the concentration or density of a substance is different on one side of a barrier compared to the other. So if we imagine the barrier is the plasma membrane of a cell, and there is a high concentration of salt inside the cell, but a low concentration of salt outside the cell, we have a concentration gradient of salt. Molecules of a substance naturally move so they can be more dispersed or further away from each other. I like to think of this like people choosing what urinal to use. Most people will go for a urinal further away from other people rather than the closest one, right? Molecules are the same way. They want their space. We mentioned that molecules that are not water-soluble can pass through the lipid bilayer, and they do this by simple diffusion. Again, these molecules are not repelled by the hydrophobic lipids of the membrane because they're not dissolved in water. But they will only pass through if they are moving across a concentration gradient. Take a look at the image on this slide. The concentration of this molecule is greater outside the cell than inside. Because molecules tend to move towards a lower concentration, this molecule will diffuse through the plasma membrane toward the lower concentration. When the concentrations are the same inside and outside the cell, the net movement will stop. Pause the video again to answer this question from Participation Assignment 1. Water also follows the same rules. Water molecules tend to move from a high concentration of water to a low concentration of water. Again, inside and outside of the cell, we have an aqueous solution. So we have water with different substances dissolved in it. In this case, water is the solvent, and the dissolved substances are the solutes. If you look at the beaker here, imagine the dotted line is a semi-permeable barrier. Water can move through this barrier, but the solute cannot. On the left side of the barrier, we have a low concentration of solute, so we have a high concentration of water. Those water molecules want to even things out. They want to move towards a lower concentration. So they will move from left to right across this barrier. Here is the same beaker after a few minutes. You can see that water moved from the left side of the beaker to the right side. Now, even though there is more water on the right side, the density of the solute is the same on either side. This is osmosis, which is a form of simple diffusion. So how do molecules that are water-soluble diffuse into or out of the cell? They need to pass through a transport protein, which again is forming a channel through the plasma membrane. In facilitated diffusion, molecules are still moving from high to low concentration, so no energy is needed to do this. Again, the molecules are just doing what they naturally do. What if the cell wants to create more of a concentration gradient instead of evening things out? It can do this through active transport, in which instead of molecules passively moving through transport proteins, they are actively pumped against their concentration gradient, so from a low to a high concentration. Because this is going against the natural movement of these molecules, this type of transport requires energy. That energy comes in the form of a molecule called ATP, which we'll talk about more when we talk about photosynthesis and cellular respiration. Now pause the video to answer this question from participation assignment 1.